Hello guys, I am Shubham from ShubhVed Tutorials. This video is about DNA replication in eukaryotes. We will discuss about initiation and elongation in this video, and in the next video we will see whole termination process. So before we start, subscribe to our channel and press the bell icon to get notified whenever new video is out. So let's get started with the video. In the previous video, we saw whole DNA replication in prokaryotes, initiation, elongation and termination. Eukaryotes also have the similar process, only the complexity is increased little bit and the proteins, okay, names of the proteins or we can say enzymes are changed. So here first step is the formation of, we know that when we see, when we say that replication has started because of formation of replication fork, right? Or we can say bubble, formation of replication bubble. Means the structure first is single stranded, right? This is a DNA. This will have a sequence. We know origin of replication, right? As we saw, a replicon model contains initiator, that is a protein, and a replicator. Replicator is the DNA sequence. Initiator will come and bind here, right? And then what happens? DNA starts to unwind. So we can see structure like this. And this is how we say that replication is initiated. So this is a basic idea, but it requires a lot of proteins and enzymes for this formation of a replication fork or a bubble. In the elongation step, what happens? Just this replication fork will move in this direction, right? As the helicase will move, unwinding the DNA, so the replication fork will be moving, continuing the whole strand replications, right? So let's see. First step is the helicase loading. Helicase loading, which takes place in the G1 phase of cell cycle. Right, G1 phase of the cell cycle. So before in helicase loading, the protein, the initiator, okay, initiator that comes and binds to the replicator DNA is the ORC. ORC, origin recognition complex. So this is the initiator. Origin recognition means this will recognize the origin of replication. Complex. This is known as ORC. So this is the initiator that will bind to the replicator. Replicator is a DNA sequence known as origin of replication. Right? Then the next proteins will come CDC6 along with ATP. Right? See, this ORC is also with one ATP. CDC is also bringing one ATP with it. Right? Then the next protein will come MCM27. This is a helicase and CDT1. So this is one subunit of a MCM. Two components of MCM27 comes in and they get joined here. See, like this. Two components are joined as well as two CDT1 are there. So this are loaded. Alright. Then what happens? ATP hydrolysis. Next step. One ATP is hydrolyzed, CDC6. The ATP that was come with the CDC6 is hydrolyzed. Then the CDT1 and CDC6 with ADP are released out. Right, are released out after helicase is successfully loaded on the DNA. We can see in this step, helicase is successfully loaded on the DNA, right, near the ORC. Still one ATP is there. As this DNA is loaded, again this ATP is hydrolyzed. Again this ATP is hydrolyzed and the helicase shifts little bit further. right? See, and this is known as successfully loading has been done. So to just understand what we can write in this step for binding, for binding of DNA, helicase on DNA, right, requires ATP hydrolysis for binding, right? This one ATP hydrolysis to place the helicase on the DNA, and the second hydrolysis is for proper location, right? Second ATP hydrolysis is for successfully loading of helicase. For that, the second ATP hydrolysis takes care. We can see. And the next step, so thus the helicase is shifted towards further, little bit further. And here it is ADP then. Okay. Again, this will get recycled for ATP. ATP will be released out and ATP will come in for again if we want to initiate or load the helicase. So this ADP is released out, ATP is inserted to recycle the process. Okay, restart the process, we can say. But restarting is not that easy. It takes place after the strands are replicated, right? When the DNA replication is completed of the strands, then only the reinitiation occurs. So this is about helicase loading, G1 phase. Now let's see helicase activation. Helicase activation, which takes place in the S phase. Okay, S phase. Now we saw that helicase is successfully loaded on the DNA. Then the next proteins come CDK, DDK in the action, right? Then CDK, DDK are the kinases. K stands for kinases. Kinases perform to phosphorylate the other proteins, right? So phosphorylation is the function of kinases. So they will CDK phosphorylates own MCM27 helicase. Okay, CDK will phosphorylate MCM27 helicases and CDK will phosphorylate this protein SLD2, SLD3. Right, after phosphorylation, they will bind to DPP11. After phosphorylation, they will bind to DPP11 and then they will enter on the DNA strand. Okay, or we can say they are recruited on the DNA strand. Then next proteins are recruited after this part. Okay, next proteins are CDC45, genes, and polymerase epsilon. We know polymerase epsilon is for leading strand, right? And this CDC45 genes are very important and they form a complex known as CMG complex. Okay, why so CMG? Because this SLD2, SLD3, and DPP11 are released out. Okay, because their work is to recruit these other proteins there. We can say like that. The function is to recruit the other proteins. See, this protein, as well as this protein are released out. Work for recruitment. So this is how we can remember this. Recruitment means entry or binding to DNA of CDC45 genes and polymerase epsilon. So we can say like that they are working for just recruitment, and then after they are recruited successfully, they will be released out. Okay. And once they are released out, the helicase is activated. You can see the strands are separated. We can see our strands are separated. Right? So they are, what we can say, they are just loading the complex, okay, loading the other protein that will form the CMG complex. That's it. Then the other proteins, polymerase. Then the polymerase come in action. Polymerase first alpha, which is a primase activity. Right? Polymerase alpha slash primase. So it synthesizes the primer. Polymerase alpha primase will synthesize the primer. Primer as well as some nucleotides will be synthesized. And then the next will come polymerase delta. Okay, polymerase delta for the lagging strand synthesis. We want to remember that polymerase epsilon comes first for the leading strand synthesis before the helicase is activated. Okay, see we saw here it comes with the CMG complex. Right. When the CMG complex is formed, polymerase epsilon is also bound to the complex. Okay. As in prokaryotes also we saw that leading strand is synthesized first, then the lagging strand. Right? Same is the case. So polymerase epsilon comes first before the helicase is activated, before the strands are separated. Right? Polymerase delta comes afterwards. After strands are separated, after primase have synthesized the primer, then the polymerase delta will come. But polymerase epsilon will come first. Right? This is the first point, and this is the second point. Okay. So this is just a picture of the CMG complex. So this is the CDC45. This is the GINS. CMG complex stands for C for CDC45, G for GINS, and M stands for MCM27, which is a helicase. Right? It is a helicase. So this is how complex is formed. And see in the before activation of the helicase, it is bound to the double strand. See both the strands are covered here. You can see both the strands are covered. But after the helicase is activated, each component will cover only one strand. So this part is covering only this strand, right? This strand, and this helicase is covering this strand. Okay, so two separate strands are now covered by the each helicase component. Previously, before activation, they are encircling both the strands. Also, we saw that G1 phase is for helicase loading. Helicase loading means MCM27, the helicase, is loaded properly on the DNA site where the strand separation will take place. Right, or we can say near the origin. Proper loading of the helicase on the double strand DNA takes place in the G1. And the S phase is for helicase activation. So in this phase, CDK levels are very important. CDK levels, kinase levels, these are increased in the S phase. Kinases are increased in the S phase. In the G1 phase, the kinases are very low. So this is a regulation of helicase loading and helicase activation in the two phases, right? So here, next, there is a term, very important term in a DNA replication in eukaryotes known as polymerase switching. Polymerase switching. What is this exactly? What is this exactly? See, we know that first primary synthesized, okay? Primary synthesized. So polymerase alpha, which is also a primase, it synthesizes the primer, right? It synthesizes the primer as well as some nucleotides are synthesized. Some more nucleotides are also added by this polymerase alpha. And then further addition of nucleotides cannot be done by these enzymes. So continue this further. This further application is done by either polymerase epsilon, that is for leading strand, or polymerase delta for lagging strand. Okay, means what? A polymerase alpha, which is a primase, which is not having that type of processivity, we can say. Okay, that is not that much powerful to continue the replication. So it will release out and the main polymerases, the polymerase delta or epsilon will come. So this is known as polymerase switching because the first work is done by one polymerase, that is polymerase alpha primase, and the next the polymerase is switched to delta and epsilon. So that is polymerase switching. So as we saw in the prokaryotes also that DNA polymerases require sliding clamp and clamp loader, right? So we can see here this original polymerase is with the sliding clamp. We know that sliding clamp increases the processivity of this DNA polymerases, right? Delta and epsilon. So this sliding clamp is also known as beta clamp. And in eukaryote, the protein that work as sliding clamp is the PCNA. PCNA. Right? PCNA ring, we say. For the sliding clamp, loader is also required. Sliding clamp loader. Okay. So that loader is the, I will write here, sliding clamp PCNA ring and clamp loader RFC. So now let's see about elongation. So elongation is nothing, just the replication fork will move forward. We will see a situation like this. Here is the fork. Okay. See, first is the helicase. This is a helicase. Okay, we can say this is a helicase. I will write here H MCM helicase. Okay. As the helicase unwinds the bonds, single strand binding protein also get bind here. Okay, this is not shown anywhere, but we have to know this. Single strand protein get bind here to prevent the strands from binding to each other again, right? So that double strand does not form again. Okay, so it is a stabilizing protein, SSB, right? This SSB in eukaryotes, the protein is known as RPA, replication protein A. And here RFC is the clamp loader, replication factor C, right? Just this will move, helicase will move in this direction and get unwinding the strands. Suppose now helicase is moved till here and these are the strands separating. Okay, here are the bonds. So this again here will come SSB, RPA, right? Like this. or inhibition of the double strands so that they cannot bind again to each other. Here are joint bonds. Correct? Here are the bonds which are joined to each other, not separated. Okay. As the helicase will unwind, go further, DNA polymerase will continue the addition of nucleotides. That's it for the elongation. So now let's take a comparison of the eukaryote and prokaryote so that we can remember the things. Prokaryotes and eukaryotes. So first is the DNA sequence origin of replication that is recognized by the initiator. Prokaryotes initiator is DNA with ATP, right? This we have to remember with ATP. ATP is common. ORC is the counterpart in eukaryote, right? First protein initiator. Second is the helicase. Helicase and helicase loader. Loader known as DNA B and C. Right. Here helicase is the MCM complex 227 and other proteins CDC 6, CDT1, then what else? CDC 45, genes. All these work for it is what we can say loading and activation. Then TDK, DDK kinases. Right? Any protein remaining? No. Then the next polymerases. No, primerases. Primerases for synthesis of primer. Here primer synthesis is done by DNA gene. Helicase loading also requires ATP hydrolysis. Here primase is polymerase alpha primase. Then next polymerase. Polymerase DNA 3 DNA polymerase 3 holoenzyme. And here polymerase epsilon for leading strand, polymerase delta for lagging strand. That's it. Okay. So this was all about initiation and elongation. Elongation, nothing, the nucleotides are added. As the helicase will unwind, the DNA polymerase will also move in this direction and go on adding the nucleotides. So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.